11:33. So with this we start, sir. Okay.
Suppose I have started the recording.
Okay, you have started. That's all right. So welcome to all the participants, to all the audience in this program. We are here once again in our lecture series on technology-based evidence for solutions in precision medicine. As we are well aware that we have been organizing this program, this lecture series since January 22 in collaboration with the Indian Institute of Technology, Jodhpur, and Jodhpur City Knowledge and Innovation Cluster, JCKIC. And in collaboration with the Center for Human Resource Development, Dr. Sarvapalli Radhakrishnan, Rajasthan Ayurveda University. This initiative, innovative initiative, was created by our visionary authorities, our Honorable Vice Chancellor of this university, Professor Dr. Abhimanyu Kumarji, and Director IIT, Professor Shantanu Chaudhary ji, and their vision has put up us in a very concrete and a very innovative path where we can find out that what our Ayurvedic medicine can be understood in terms of the technological aspect. For this purpose, university and IIT has been heading towards under a very prestigious project, IUTech project, which is working in the field of various explanations, various inferences which has been explained in Ayurveda, and to explore them on the technological platform. So, for this purpose, the Ministry of Ayush has sanctioned a Center of Excellence, IUTech Center of Excellence in IIT Jodhpur, which is being established in IIT Technology in the fast manner, and soon it will be established here. And we are working under this umbrella and the ambit of this center. And this lecture series is also based upon that platform. And as I said, that we are holding a lecture series on technology-based evidences for Ayurvedic solutions in precision health. In this regard, already we have organized 2 lectures, one in January and another one in April. And today we are organizing 3rd lecture, which is being delivered by Dr. Raviraj, Vankayala, an assistant professor at Department of Bioscience and Bioengineering, IIT Jodhpur. This lecture named Theranostic Nano-medicines, translation from bench to bedside beyond. As we know, the science is growing very fast and various innovations are being taken up. And now the scenario has, the development has gone up to the level of nanomedicine now. And in this regard, IIT has been working very efficiently. And Dr. Raviraj Vankayala has a very commanding knowledge in this sector. And today he is going to share us with his experience and knowledge with all this. I hope the Ayurveda fraternity across India and working in various institutions will certainly be very much benefited by this knowledge, by this new window of knowledge. Before I start this lecture, I would like to introduce Raviraj Vankayala ji. Raviraj Vankayala ji having a very keen interest in the field of nanomedicine, biophotonics, theranostics, and drug delivery. He has done PhD degree in chemistry from National Tsinghua University, Taiwan in 2012, and he has done his postdoctoral studies in National Tsinghua University 2015 and University of California, Riverside 2017, USA. He worked as a Chief Technology Officer for Red Optics LLC from 2017 to 2019 for the development of biomimetic nanoconstructs for imaging and therapeutic applications of cancer and vascular diseases. He has published around 45 papers so far in various reputed journals, including Advanced Materials, Advanced Functional Materials, Ange Chemi, Biomaterials, and Small. He is a recipient of Young Scientist Award by Andhra Pradesh Academy of Sciences for the year 2016 and secured prestigious Ramalinga Swamy Biomedical Fellowship for the year 2018-2019. He is also the recipient of IIT Jodhpur Research Excellency Award 2021 in the area of nanobiotechnology. He served as a guest editor for MDPI Bioengineering, MDPI Nanomaterials, and Frontiers in Nanotechnology journals. He is currently serving as a review editor for Frontiers in Bioengineering and Biotechnology, Frontiers in Molecular Biosciences, Frontiers in Chemistry, and Frontiers in Drug Delivery journals. Sir, I, along with my co-organizing member, Dr. Hari Singhal, I cordially welcome you on this platform, sir, and hopefully I will be enjoying this lecture throughout the session. I request you to please continue with your lecture now, sir, onwards, and benefit our audience. Thank you very much, sir. Dr. Raviraj, Vankayala, please.
So, Dr. Rakesh, can you please confirm to me whether the screen is visible in slideshow mode or not?
Yeah. Your slides are visible, sir.
Okay.
Sir,
okay.
Sir.
So, first of all, thank you so much, Dr. Rakesh and his colleagues from Ayurveda University for having me for this prestigious lecture series. Yeah, thank you for the invite. So, the title of my talk is Theranostic Nano-medicines, Translation from Bench to Bedside and Beyond. So, most of my talk primarily focuses on cancer. As you can see, cancer is one of the deadliest diseases which really affects the survival rates of humans. And if you look at the number of cancer cases, or if you start looking at the global scenario of cancer, so basically for the next 20 years, like by 2040, there is a huge increase in the number of new cancer cases according to the prediction. So this is across all different continents ranging from Asia, Europe, North America, Latin America, Africa, and Oceania. And if you look at the number of new cancer cases in 2020 across males and females, so in males, lung cancer and prostate cancer being the most predominant one, and in females, breast cancer as well as the colorectal cancer has been the most predominant one based on the statistics from 2020. And nanomedicine, it is an interdisciplinary subject. So it basically integrates biology, it integrates nanotechnology or material science, and it also integrates chemistry. And disease and ill health both are caused by the damage at the molecular level as well as at the cellular level. And if you look at most of the diagnostic tools as well as therapeutic technologies, most of them are imprecise and they are not really effective. So what nanomedicine can actually do is it can monitor, it can repair, it can construct, it can also control the human biological systems at a molecular level using various kinds of engineered nanostructures. So talking about a little bit more on the beauty of nanotechnology in oncology research. So most of the traditional molecular drugs, they can damage both malignant cells as well as normal cells, and thus a drug delivery system can selectively target a malignant tumors is much needed. And the advantages of drug delivery systems by these nanoparticles could be because these nanoparticles can offer a very good encapsulation efficiency, and it can also have an excellent packaging protection with respect to the molecular drug. And comparatively, compared to the traditional molecular drugs, the less amount of drug is actually required for the treatment process, and it can also prolong the circulation half-life of the molecular drugs, and it can also achieve excellent molecular specificity or molecular targeting, and it can accumulate at the target tumor sites. Either it could be due to the active targeting or the passive targeting, and overall, with the help of these drug delivery systems, you can achieve very minimal side effects. Now let us look at the impact of different kinds of nanoparticle delivery to the tumors. So as everyone knows that a term nanoparticle is primarily composed of its, the most important things are like the physical properties. It has its structure where you can talk about its chemical composition, and since nanotechnology can offer diverse surface chemistry, you can always modify with different kinds of targeting ligands, small molecules. You can also modify with antibodies, aptamers, different kinds of proteins, and etc. So the first thing, looking at the physical property, the size of the nanoparticle, the morphology of the nanoparticle, and the surface charge of the nanoparticle, that is pretty much the zeta potential, and the porosity, elasticity, and stiffness, and electronic, magnetic, and other optical properties. All these are very, very important, and these are the physical properties, and these can be tuned with the help of with the help of chemistry and with the help of material science. You can actually tune all these properties depending on what kind of application you wanted to utilize them for. And coming to the structural aspects of these nanoparticles, that is primarily we are looking at the chemical composition. So where you can see you have liposomes, which are composed of both natural and synthetic lipids, and you have polymeric nanoparticles, which are mostly, they are biodegradable, and then you have micelles, you have dendrimers, and there are a lot of different kinds of biomimetic nanomaterials as well, such as viral nanoparticles and red blood cell derived nanoparticles, and etc. And in addition to that, you also have metal and metal derived nanoparticles, for example, gold nanoparticles, silver, and other metallic nanoparticles, and carbon based materials. And there is also another class of nanomaterials called hybrid nanoparticles, where you can combine the efficacy, or you can combine the properties of two different nanoparticles and make a hybrid system. And as I told you, the targeting properties are also important, where you will be able to conjugate different kinds of small molecules, antibodies, aptamers, and nucleic acids, and proteins, peptides, and other molecules, and etc. Now, if you design the nanoparticle in this way, how the nanoparticle properties will actually affect the delivery to the tumor. So once you inject a nano-based formulation into the patient or into a small animal, the very first thing, usually these nanoparticles are injected through the systemic injection, through the systemic circulation, or through the intravenous injection. And the first thing it encounters is it enters into the bloodstream, where it can encounter a lot of different kinds of serum proteins. So these serum proteins tend to absorb onto the surface of the nanoparticles, and then over the period of time, you can actually monitor the plasma concentration, where how the nanoparticle lifetime, whether it is degraded by any other kind of macrophages which are present in the reticuloendothelial system, or how the biodistribution varies in most of the biological organs, you can actually monitor. And then once it reaches to the tumor site, and then it can have, as I told you, it can have either active targeting, or it can also have passive targeting. So active targeting means with the help of a specific receptors which are overexpressed on the cancer cells, you can always modify your nanoparticles with the antibody and then you can actually the tumor cell can bind to that particular receptor which is being overexpressed on it, and then eventually it can be endocytosed within the tumor cell. And then you always have the passive targeting where the vascular extravasation can take place because of the leaky vasculature of the tumor cells. These nanoparticles, if you can tune the nanoparticle size with an, like, let's say 100 to 150 nanometer, and then it can actually undergo a very good vascular extravasation through the EPR effect. And this EPR effect is called as enhanced permeability and retention effect. And once you have a nanoparticle based drug delivery system, and then and once it is injected into the body, and once it gets biodistributed through different parts of the biological system, and once it also reaches to the tumor cell, then you need a stimuli. You need a different kinds of stimuli so that the drug molecules which are encapsulated into these nanoparticles can trigger the release over a period of time. And these stimuli could be either an external stimulus or it could be an internal stimulus. Some of the examples of the stimuli could be the light, it could be the ultrasound, or it could be pH, it could be temperature, and etc. Now let us talk about the term called multifunctional nanodiagnostics, because the title of my talk is primarily on diagnostic nanomedicine. So first, let me introduce what exactly diagnostics is. So this particular term, diagnostics, is a kind of a buzzword right now. And so basically, diagnostics is a combination of therapeutics and diagnostics. So it is derived from the word therapeutics as well as the word diagnostics. And both of these modalities are integrated into a single, a multifunctional nanoparticle system. So what kind of advantages that these diagnostics can give in comparison to that of the individual modalities? The first thing is it can provide a very good synergism, that you can have two different modalities together, and then you can have multifunctionality, and then you can also have localized targeting. You can have a long-term effect.
बाकी सब लोग माइक बंद। हरीश जी एक सेकंड, जस्ट अ मिनट। हरीश जी बाकी सबको म्यूट कर दीजिए। डॉक्टर साहब
हेलो,
हां, एक जस्ट अ सेकंड सर, पीछे से नॉइज हो रहा है सर, सॉरी टू डिस्टर्ब यू। सबको एक एक दो हरीश जी चित्रा भाई बताओ उसने क्या कहा। हेलो,
यस सर।
डॉक्टर साहब अपनी आवाज बंद करें प्लीज, माइक म्यूट करें।
यस सर, यस सर।
हाँ हरीश जी, बाकी सबको म्यूट करते हैं, खाली रवि जी को ऑन रखें, है ना।
ओके।
सभी लोग प्लीज ध्यान रखें अपना, कोई भी बीच में जो है अपने माइक म्यूट रखें, ठीक है। डॉक्टर पवन जी, ठीक है। डॉक्टर पवन यादव प्लीज अपना माइक ऑफ रखें, ठीक है। यस।
And hello, am I audible?
Yes, sir, you are audible.
Okay, yeah. So these kind of nanoparticles can provide synergism, multifunctionality. It can also achieve localized targeting, and it can also have long-term effects. And as I mentioned in the previous slide, these nanoparticles can be responsive to certain kinds of stimulus as well, so that the therapeutic payload or the drug payload can be released. So what exactly a multifunctional nanoparticle consists of? The ideal multifunctional nanoparticle actually consists of, like, let's say if this is a 100 nanometer a multifunctional nanoparticle system, so you will have an opportunity to encapsulate different kinds of drug molecules into it, and you can also modify the surface with different kinds of targeting ligands that could be aptamers, monoclonal antibodies, and etc. And you can also conjugate the surface of the nanoparticle with different kinds of a linker or spacer molecules as well, and you can attach different kinds of genes, and in order to have the imaging modality, you can also conjugate these nanoparticles with the help of the fluorescent dyes as well. And overall, now we are gradually shifting from the conventional one-size-fits-all approach to the holistic, personalized medicine-based approach with the development of multifunctional nanodiagnostics. Now, I would like you to take you through the research publication statistics in this emerging field. So this particular data has been pulled out from ISI Web of Science. And if we have a keyword of diagnostics, and if we look at the publication statistics as well as the citation statistics in the field of nanodiagnostics, there are almost 10,000 publications has been come up. So then you can understand how important is this field is, and how this field is actually emerging over the period of time, over the past 2 decades. And specifically, if you narrow down your search to nanodiagnostics, there are almost 906 publications have been coming up over the past decade, over the past 10 years. So currently, there are a lot of clinical diagnostic modalities. So here I'm going to classify the clinical diagnostic modalities into 2 categories. One is optical modalities, which is represented in the red color, and the other one is non-optical modalities, which is represented in blue color. So both these optical modalities as well as non-optical modalities are classified in terms of its penetration depth and resolution. So as you can see that the non-optical modalities such as X-ray, computed tomography, PET, or MRI or ultrasound, although they offer a very good penetration depth, but their resolution is compromised. Their resolution is at a level of 10,000 microns. But if you look at certain optical modalities such as photocaustic imaging, optical coherence tomography, as well as near-infrared fluorescence imaging, although they can provide a reasonably good penetration depth, but they can also achieve a very good resolution. So we have ample opportunities in terms of nanotechnology derived photocaustic agents or nanotechnology or different kinds of nanoparticles to mediate optical coherence tomography imaging, or also different kinds of nanoparticles to mediate near-infrared fluorescence imaging. And if you look at the therapeutic modalities or the treatment modalities, I think the most common treatment modality is the surgery, which is kind of a non-invasive technique, which is kind of an invasive technique. And then you have chemotherapy, where different kinds of drug molecules are used, and those drug molecules could be Taxol, doxorubicin, cisplatin, depending on what kind of a tumor you're treating. And the majority of the drawbacks of these drug molecules is that they can, over the period of time, over the repeated treatments of chemotherapy, they can achieve tumor recurrence. I mean, they suffer from the multi-drug resistance problem, and because of that multi-drug resistance issue, the chemotherapy will no longer become effective, and then the tumor can be reoccurred. And then you also have a radiotherapy, or sometimes radiotherapy often uses in combination with chemotherapy as well. But the drawback of radiotherapy is that it uses ionizing radiation to kill malignant tissues, and radiotherapy is non-specific, and it also poses a high risk of damage to the normal tissues as well. And the other less prominent modalities are nothing but gene therapy, where you can repair or modify a gene, and most of the gene delivery vectors which are currently available, they are not effective, and in terms of it, because they exhibit a very poor gene therapeutic efficacy, and they are also toxic. And then you have boron neutron capture therapy, which is more popular in the Southeast Asian countries, where they use a boron enriched reagent along with a combination of a neutron irradiation to produce gamma rays and the alpha particles. And these gamma rays and alpha particles can actually damage the tumor tissues very effectively. And the last one is phototherapy. Phototherapy is, again, it is one of the minimally invasive treatment modality. And what it primarily requires is it needs light, oxygen, as well as a photosensitizing dye molecule. So throughout my presentation, for the next couple of slides, I'll be focusing on gene therapy, boron neutron capture therapy, and phototherapy. Now, let us look at some examples of organic photosensitizer mediated photodynamic therapy. So here I'm talking about a phototherapy. So ideally, in the clinical setting, so when a patient comes to the hospital, you can inject a photosensitizing molecule. Like let's say a patient has a tumor, you can inject the photosensitizing drug molecule, and over a period of time, like let's say after 24 hours or 48 hours, the photosensitizer will concentrate to the tumor area, and then once you irradiate the tumor area with the help of light, the tumor can be selectively destroyed. So these are the different 4 steps involved in organic photosensitizer mediated photodynamic therapy. But if you look at the molecular mechanism or the photochemical mechanism involved in these photodynamic therapies, that once you have a photosensitizing molecule and you irradiate the photosensitizing molecule with the help of light, you kick out the electrons from the photosensitizing molecules so that the electrons can jump to the upper excited states, and from those upper excited states, it can either relax back to the singlet as well as the triplet states with the help of different kinds of photochemical processes such as intersystem crossing or internal conversion to induce fluorescence as well as phosphorescence. And that phosphorescence could be with the help of the molecular oxygen, where it can produce a lot of reactive oxygen species, type 1 and type 2 reactive oxygen species, which can induce apoptosis or necrosis, which can contribute to the cellular death. And most of the commonly used photosensitizing molecules are aminolevulinic acid. So this aminolevulinic acid, which is activated by 275 nanometer UV light, and then we also have photoferrin, which is activated by 550 nanometer light, and then you also have Foscan, which is activated by 650 nanometer light. So these 3 are the most popular drug molecules which are FDA approved and currently used in photodynamic treatments, and mostly they are used for treating the surface tumors because, as you can see, they are mostly activated by UV or visible light. Now, if you look at the biological spectrum, in addition to the UV light and in addition to the visible light, you also have ample opportunities to be explored in the near infrared region. So, and most importantly, this particular near infrared region is called as a biological window because it can give you reduced scattering, and it can also have a very high tissue transparency because the major components of the blood is the hemoglobin and the oxyhemoglobin, which absorbs in the visible region. And even you have the major component of the skin, which is melanin, which also absorbs in the visible region. So near infrared region has a very high tissue transparency, of course, with a very minimal scattering. And not only it can have a reduced scattering and a high tissue transparency, it can also offer deeper tissue penetration. So what is the advantage of having a deeper tissue penetration is that it can treat tumors which are deeply buried in the tissue. Like for example, not just with the skin tumors or the skin cancers, which I'm commonly talking about. It can also provide an opportunity to treat like let's say brain tumors, which are like more complicated. Now, here comes the beauty of nanotechnology. I'll for the next couple of slides, I'll be talking about how different kinds of nanoparticles can be used to, where the diagnostics as well as therapeutic components can be integrated in different kinds of multifunctional systems to treat various tumors. So the first example I would like to talk about is metallic nanoparticle. I think all of you are aware about metallic nanoparticle because gold nanoparticle, silver nanoparticle, these are very common now, and they have diverse applications ranging from environment to healthcare to energy related applications. And these metallic nanoparticles, as you know, they come under the class of plasmonic material. They come under the class of plasmonic nanoparticles because once you irradiate these metallic nanoparticles with the light, they produce localized surface plasmon resonance. And once they are irradiated with the help of light, and these nanoparticles can also physically absorb because of its surface phenomenon, the molecular oxygen can be efficiently absorbed to the surface of the metallic nanoparticle. And once you irradiate with the help of a light, there undergoes an energy transfer. The plasmonic energy undergoes a transfer to the adsorbed molecular oxygen, and then it kicks out the singlet oxygen phosphorescence emission. And this particular demonstration shows that these metal nanoparticles can sensitize the formation of singlet oxygen upon direct photoexcitation. So that means these metallic nanoparticles can be used directly for the treatment of patients, for the treatment of tumors, in order to kill the cancer cells without having any kind of drug molecules. And why these metal nanoparticles are advantageous? Because it has a very good photostability when compared to the conventional drug molecules, and it is also resistant to the enzymatic degradation, and it also it can also have a very good light absorbing capability when compared to the conventional molecular drug, and it can be used as a photosensitizer for photodynamic therapeutic applications. I'm not going deep into the chemistry part, but I'll try to bring out the essence involved or the mechanism involved in most of these systems. And again, if you remember, if you recall one of my initial slide, I have mentioned that with the help of nanotechnology, you can tune the size of the nanoparticle, and you can also tune the morphology of the nanoparticle depending on what kind of application you're looking at. Like for example, with respect to the morphology, you can come up with a nanorod, you can come up with a nanosphere, you can come up with a nanocube, you can come up with a triangular plate, nano-triangular plate. So different morphologies of nanoparticles can be synthesized. So once you change the morphology of these nanoparticles, their optical properties can also be tuned. So because these nanoparticles are composed of different kinds of crystalline facets, and different crystalline facets have different kinds of plasmonic surface energy levels, and they absorb at different wavelengths. So most of these nanoparticles, they can absorb in the near infrared region. So that means it gives an opportunity for us to develop different kinds of morphologies of nanoparticles where you can irradiate with a near infrared light, not the UV light or the visible light. I'm repeating again, it is with the infrared light where it can offer deeper tissue penetration. And that deeper tissue penetration is highly advantageous to treat complicated tumors, such as like deeply seated tumors, whether it could be ovarian tumors or brain tumors, and etc. And this singlet oxygen sensitization depends on the morphology of the nanoparticle, where the selective molecular oxygen binding can occur on a particular crystalline facet. And the infrared light excitable PDT agents are rare, and especially among all the morphologies tested, the gold nanorod can be turned out to be as the best or the superior photodynamic therapeutic agent. So this is just the proof of concept study, which was published in Small in 2014, where we photo-excited gold nanorods with the help of a near-infrared light, where it can produce heat and it can also produce ROS. So this heat and the reactive oxygen species together can deliver phototherapeutic effects. And if you irradiate with an alternate wavelength other than the near-infrared light, these gold nanorods can also produce a very good fluorescence emission, a one-photon excitation process with the help of the one-photon excitation process. And this particular fluorescence emission can be utilized for fluorescence imaging for the diagnostic purposes. So that means it provides an ample opportunity to monitor the therapeutic outcome of a disease process with the help of the imaging modality. So these gold nanorods could be termed as a diagnostic agent or a multifunctional nanodiagnostic agent. And this is just a proof of concept study where we have done a series of in vitro experiments as well as in vivo experiments in melanoma tumors to show the therapeutic efficacy of gold nanorods as NAR light activatable phototherapeutic agent. And then we went a step ahead, and then we try to synthesize a different kinds of a morphology of gold nanoparticles, which is called as gold nano-echinus. And this gold nano-echinus, so echinus is a sea urchin. So I think everyone, most of you are aware of sea urchin. So this sea urchin, this echinus looks like a sea urchin, and it has multiple nanorods which are protruded from a single nanoparticle. So can you imagine if a single gold nanorod could be very effective in terms of treating tumors? Then if you can design gold nanoakinase heterostructure where multiple nanorods are being protruded from a single nanoparticle, then you have a very broad absorption covering both the biological windows. So it gives an ample opportunity to cover the entire biological spectrum. And most importantly, these nanomaterials, they can also have a very good light absorbing capability. So what is the advantage of having a very good light absorbing capability is that if you use a very tiny amount of nanoparticle, and if that tiny amount of nanoparticle can be excited with the light, it should be good enough in order to mediate the superior therapeutic effects. So you can minimize the toxicity which is coming from the material. So you can inject a very tiny amount, you can low down the amount when it comes to treating for the patient. And as I told you that this particular gold nanoakinase, it can produce photodynamic therapy both in the first biological window, which is typically 650 to 950 nanometer, and then it can also produce photodynamic therapy in the second biological window as well, where it typically consists of 1000 to 1350 nanometer. And this work has been, and this particular work, taken further, we have done a series of in vitro and in vivo experiments using these gold nanoakinase in the HeLa cells in the in vitro, and also in the melanoma tumors in the in vivo, using both the biological windows. And when compared to the conventional chemotherapeutic approach, we have achieved a very significant phototherapeutic efficiencies with the help of gold nanoakinase. And this work has been published in 2014 in Advanced Materials Journal, and it was also highlighted as a cover picture in that particular journal back then. And there are many other examples of nanomaterial mediated photodynamic therapeutic agents, and we have summarized the nanomaterial mediated photodynamic therapy in an extensive review paper, which was published again in Advanced Materials Journal in 2018. So if any of you have interest in this particular field, if you want to read more, you can always go back and refer to this particular review paper, where it covers the entire spectrum of nanomaterials with different sizes and what kind of targeting moieties it has, and what kind of irradiation conditions it produces, and what could be the efficacy of the singlet oxygen generation capability. So overall, as I told you, if you talk about the energy diagram of the nanomaterial, these plasmonic materials are composed of localized surface plasmon resonances, right? So if you excite a nanomaterial with the help of light, so then you excite the electrons to the upper excited state, you excite the localized surface plasmons to the upper excited state, to the singlet state. So from there, it can relax back to the ground state with the help of fluorescence, or it can also produce a non-radiative decay in the form of heat. And then it can also, from upper singlet state, it can also undergo intersystem crossing to triplet state. And in the triplet state, it can undergo energy transfer process with the help of the adsorbed molecular oxygen to produce this kind of a singlet oxygen, where it can trigger the photodynamic therapeutic effects in the form of a phosphorescence emission. So this is basically the energy diagram for the nanomaterials. So now, after looking at a couple of examples of different kinds of nanoparticle or nanomaterial mediated photodynamic therapy, now I'm slowly jumping into the gene therapeutic approaches. So as you people are aware, there are different kinds of challenges in gene therapy, and you have like, for example, if you look at different kinds of gene delivery vectors, you can there is a microinjection procedure, and there are different kinds of a chemical-based gene delivery vectors, and there is electroporation, and there is also with the help of a mechanical and electrical simulation, where you can deliver the respective genes to the target cells. But most of these processes, in the process of achieving superior delivery efficiencies, they also produce a lot of toxicity. So the major challenges in gene therapy is to design an ideal gene delivery vector which can have a very good intracellular uptake, with a very minimal cytotoxicity, and at the same time it should also produce a very good gene delivery efficiency, and it should also overcome the endosomal and lysosomal degradation, and it should also facilitate a flexible binding between the DNA as well as the nanocarrier. So one such example I would like to highlight is that we have developed a lipid iron carbon nanoparticle based composite system, where these iron carbon nanoparticles are composed of you have an metal core which is of iron core, and then surrounding the metal core you have a graphene layers or the carbon shell surrounding to it. So this is a very homogeneous morphology which has a size distribution of like let's say 25 to 30 nanometer, and these nanoparticles can be integrated into a lipid bilayer where it can form this kind of a lipid nanoparticle composite system. And if that lipid is composed of a cationic lipid, then you can attach polyanionic moieties onto the surface of the lipid carbon nanoparticle based composite with the help of electrostatic interaction and facilitate the gene delivery efficacy. And we have looked at the intracellular uptake as well as the gene transfection efficiency, both in the in vitro, in the HeLa, the cervical cancer cells, as well as in the zebrafish model. And then we found out that these nanoparticles, like the lipid carbon based nanoparticles, they turned out to be a very good gene delivery carrier, where it can facilitate a tight binding, but yet it can also become detachable, where the plasmid DNA can be effectively released when compared to the other gene delivery vectors. And yet, if you look at the cytotoxicity, they are at a much lower scale in terms of its cytotoxicity. And this work has been published in the year 2014 in Biomaterials. And as I told you in the initial, one of my initial slides, that I'll be focusing on phototherapy, gene therapy, and also boron neutron capture therapy. So boron neutron capture therapy is one of the very promising treatment modality, where this particular BNCT is clinically used for treating brain tumors and also the head and neck tumors. And why in particular this boron neutron capture therapy modality is used for treating the glioblastoma or the brain tumors is because this particular modality doesn't have any kind of a penetration depth issue, because the neutrons can actually achieve a very good penetration, and it can deeply dive into the brain where the tumors are located. And how do you generate these boron neutron capture therapy agents? Is what is the mechanism involved is that, so you can develop a boron enriched reagent, and once you have the boron enriched reagent, which is bombarded with a beam of dermal neutrons, and then it can emit alpha particles, and these alpha particles can kill the cancer cells. It can damage the cell membrane, and it can induce the apoptosis or the necrosis process, where it can eventually kill the tumors. And there are different kinds of boron neutron captured therapy agents have been developed for different kinds of applications, for different kinds of for treating different kinds of tumors. There is a liposomal polyhedral carborane. There is boron encapsulated carbon nanoparticle, and there are two different kinds of molecular drugs. One is a boron phenylalanine, and the other one is BSH. But all these different kinds of agents which I have mentioned right now, they suffer from very poor tumor to blood boron ratio, and they also lack of enough boron 10 isotope in its nanoparticle. So in our strategy, we have developed the boron 10 based nanoparticles for boron neutron capture therapy of brain tumors, where we have taken boron 10 nanoparticles and we have coated the boron 10 nanoparticles with a layer of APTES, where you can get this kind of a core shell kind of a structure. And once you have the silica in it, it can achieve a good biocompatibility, and it can also offer you some certain porosity where you can entrap or where you can load different kinds of small molecules. So in order to make this as a diagnostic agent, we need a imaging modality. So in order to have an imaging modality, we use a conventional gadolinium DTPA complex encapsulated into the pores of the silica. And then we also targeted the whole system with RGDK FIT3, which can target the alpha V beta 3 integrin, which is overexpressed on the glioblastoma tumors. And we have done a series of in vitro and in vivo studies as well. Like in the in vitro, you can very clearly see that as you increase the concentration of these nanoparticles along with the neutron irradiation, they achieve a very significant amounts of cell death when compared to the conventional boron phenylalanine with the help of neutron irradiation process. And in the in vivo mice model, we have developed the brain tumor model in the in vivo, and then we have administered these nanoparticles through systemic injection, where with the help of MRI, we were able to locate where the tumor is, and we were also able to monitor the therapeutic outcome of the boron neutron capture therapy with the help of the MRI imaging modality. As you can see from the histopathology data, the nanoparticles with neutron irradiation has the lowest diameter of the tumors, has the lowest volume of the tumors when compared to the other treatment groups, and it also improves the survival rate much effectively. And another example I'll be talking about is the lanthanum hexaboride nanocube. This is again a diagnostic nanosystem, and this particular diagnostic, this particular lanthanum hexaboride nanocube can actually mediate the photodynamic therapy in the NAR2B wavelength region, that is beyond 1350 nanometer. So that means it produces photodynamic therapy upon excitation with 1550 nanometer wavelength. So that means when compared to the UV visible and the infrared light in the first biological window, so nanomaterials which are activated at NAR2B wavelength could combat deeply seated tumors much more effectively. So in this particular system, we have taken lung tumor, the non-small cell lung cancer, and then we have the anti-EGFR, which overexpressed on non-small cell lung tumors are modified to the surface of the lanthanum hexaboride nanoparticles. And then we have performed a series of in vitro and in vivo studies where we developed the proof of concept to show that these nanoparticles can actually treat the lung tumors very, very effectively. And until now, I have gone through the phototherapy, I have gone through the gene therapeutic modality, I have also gone through a little bit of boron neutron capture therapy as well. Now, and I have taken you through different kinds of nanoparticle based systems, and these are mostly the hard nanoparticle based systems which I have taken you through. Now I will provide some particular strategy, nanoparticle biomimicry strategy, where you can improve the circulation of these nanoparticles. So how you can improve the nanoparticle circulation is either you can pegylate the nanoparticle, you can always do the pegylation reaction to the surface of the nanoparticle, and you can also attach a self-peptide. And the third strategy could be you can coat or camouflage the nanoparticle with the help of a RBC membrane or a leukocyte membrane or an exosome. So these are the different kinds of nanoparticle biomimicry approaches to achieve prolonged circulation. And if you look at the FDA approved drugs, one of the important drug or a dye molecule I would like to highlight is indocyanine green. Indocyanine green is the only near infrared approved dye molecule, and it is approved for imaging applications, especially for ophthalmological applications. And this indocyanine green, it is a benzoindotricarbocyanine which has hydrophilic as lipophilic moiety. It has a very poor, although it absorbs and emits in the infrared region, it has a very poor plasma half-life, and which is again non-specific. So in order to overcome the limitations of these indocyanine green molecules, we have developed a nano platform, a biomimetic nano platform, which is composed of red blood cells. And in this particular platform, we take the whole blood and we subject that to the hypertonic treatment where we deplete the hemoglobin and we end up with the erythrocyte ghost. And this micron-sized erythrocyte ghost is mechanically manipulated to bring it to the nanocyte. And then the indocyanin green molecules can be encapsulated to make it this kind of a near-infrared erythrocyte mimicking transducer. And the advantages of this kind of a biomimetic nano platform is that it can have a very good circulation time. And these constructs, as these constructs are fabricated autologously, they can also be compatible from various blood types. And this could be potentially non-toxic and non-immunogenic. And the diameter can be tuned depending on your application, ranging from 100 nanometer to 4 micron. Like if you're looking at a micron-sized particles, you can always use for treating vascular diseases. And the optical properties or the optical properties can be tuned because you can load with different kinds of a drug or dye molecules, and the dye or the drug content can also be controlled very effectively. And this is just looking at the set of microscopic characterization for the micron-sized particles as well as the particles. And it also has a very good.
एक बार मैं लिख रहा था उसमें।
अच्छा गोविंद गुप्ता जी प्लीज गोविंद गुप्ता जी प्लीज माइक बंद ऑफ करें।
And it also,
sir, please.
It also, as you can see, that these nanoparticles can also, achieve a very good circulation time, a prolonged circulation time, and where it improves the bioavailability of ICG. And not only that, these nanoparticles can be activated by infrared light, and it can also produce heat as well as reactive oxygen species, where the ICG can be excited, and then it can produce a very good phototherapy. And this work has been, so several papers have been published using this particular nanotechnology. And we also, further, we can also conjugate these nanoparticles with different kinds of, targeting moieties so that it can achieve a very good molecular targeting. And this is one example where it can show a targeted near-infrared fluorescence imaging where it can lighten up the ovarian tumor. So these nanoparticles are targeted with the folate moieties, which are overexpressed by the ovarian tumors, and they specifically go and bind to the ovarian tumors and they lighten up there. And after looking at different kinds of the hard nanoparticle based technologies as well as the soft nanoparticle based technologies, how this desert inspired nanotechnology can be effectively used for medicine. So we live in Rajasthan and we have a lot of flora and fauna in and around us, and there are a lot of animal species which are specific to the Thar desert here in Rajasthan. So how we can utilize the desert inspired nanotechnology in the field of medicine. So a couple of examples I would like to point out is, as you all know, that camel milk has a lot of medicinal benefits. It curbs the hunger. It has a very good anti-diabetic properties, and it also has a very good wound healing capabilities and etc. So there are a lot of medicinal benefits which are associated with camel milk. And you also have the caltropis, which is popularly, which is seen everywhere here. And you also have lantana camara, which is again has a very good antibacterial activity, anti-fungal activity, and anti-inflammatory activity, and etc. So how these medicinal plants, or how these the desert inspired the species can actually inspire the nanotechnologies, where it can be applied for for treating or diagnosing different kinds of diseases. So the first example is, so there are some literature reports where the caltropis, the procrea, which is a species of a flowering plant, which is again native to the Thar desert here. And the fruits, the green fruits which you see here, it contains a toxic milky sap, which is extremely bitter and turns into gluey coating, which is resistant to soap. And these the leaves or the milky sap or the fruit which is present in it can be mixed with a copper acetate, and then the nanoparticles can be prepared. The copper nanoparticles can be prepared using this. So this, you call it as a biosynthesis, because you prepare these nanoparticles from the biological extracts, which are typically the plant extracts, and then you further test the activity of these nanoparticles, which are synthesized by the plant extract in different kinds of cancer cells, to see how well it can treat, or how well it can affect the proliferation. And in my lab here, we have taken up this kind of a research where we started exploring, where we started utilizing the properties of camel milk. So we have taken the camel milk, and then we have condensed the camel milk into a hydrothermal oven at 180 degree Celsius for 3 hours. And then we obtain a carbon quantum dot. And these carbon quantum dots are subjected to different kinds of different steps of purification process, where you finally obtain a nicely powdered carbon quantum dot, which can absorb very well in the UV region, UV visible region. And not only that, it can also give you a very good fluorescence emission. So these carbon dots are derived from these natural sources, and these carbon dots, or the carbon quantum dots, they are very tiny in size, and they have a huge potential for drug delivery applications, or these carbon dots can be directly tested in terms of its antibacterial activity or anti-tumor activities, and etc. And as I told you that these carbon dots, which are prepared from this camel milk, they give you a very good fluorescence in the blue wavelength region, but because of its surface defects which are present on the carbon dots, they also produce an excitation wavelength dependent fluorescence emission. And so that means if you tune the excitation wavelength of carbon dots, the fluorescence emission can also be tuned to the longer wavelengths. So that means it gives an ample opportunity for us to develop different kinds of nanoparticles derived from these natural sources, and it can be utilized for different kinds of diagnostic applications where we'll be able to tackle different kinds of diseases. And these carbon dots are also used for metal ion sensing application. And again, this is just a proof of concept study where we have explored these carbon dots to sense different metal ions. And out of all the metal ions tested, we were able to sense the manganese ions very effectively at a wide range of concentrations. And the other aspect, the second aspect in that is we also prepared a carbon quantum dot from the lantana camera extract as well. And it also produced a very good fluorescence emission, the blue color fluorescence emission. And it also shows a very good excitation wavelength dependent, the fluorescence. So this fluorescence can be tuned depending on the excitation wavelength you use. So overall, this is just the starting of the work, where we can come up with different kinds of opportunities to utilize the flora and fauna of the medicinal plants which are present in and around here, and we can engineer the nanostructures very effectively from these, and they can be used for treating different kinds of diseases. And overall, I would like to conclude or summarize my talk here. So whatever nanoparticles which I talked in today's talk, whatever I projected, they have a lot of applications in either it could be a fluorescence guided cancer surgery, or these materials can also be used as a very good antibacterial agent where you can tackle the infections. And if you are looking at some certain kind of a micron size structure, those could be very effective in treating the vascular diseases such as port wine stains. And nanoparticles can also be can also have a lot of implications in getting new antivirals, maybe including COVID-19. And it also has a lot of implications in diabetes as well as the thrombolytic disease as well. So overall, the field of nanotechnology and the desert ecosystem can together can provide the bioengineering solutions to healthcare and environment. And this is where actually the knowledge of Ayurveda can also be integrated into it, and more effective technological advancements or effective bioengineering solutions can be provided to both healthcare as well as environment. So finally, I would like to acknowledge my research group, the Nano-Med laboratory, and all the lab members here. And also I would acknowledge all my research collaborators as well as the funding agencies. And thank you so much. And I would like to take up the questions.
Thank you, Dr. Ravi, sir. It has been a very comprehensive analysis, and your experience which you have shared with us. I think this is quite a new window. This is quite a new knowledge as far as our Ayurvedic fraternity is concerned, because you people hail from the technology side, and though technology includes each and everything everywhere, we see that technology has its roots in everywhere. Ayurveda also is one of them, and our Ayurveda as well as the modern science, modern health science, both sciences have a very much, you can say, the functionality, the functionality of medicine in both the sciences works upon on the basis of the nano-medicine technology, as you have explained. And the need is to identify the mode of action, the mode of the functioning of these medicines at the nano level is the need of the hour. And I think if we get through this phenomena, then only we would be able to understand the mechanism of treating, whether it is by virtue of Ayurvedic system of medicine or by virtue of modern system of medicine. If we can understand these things, we would be very much efficiently able to make the human society free of diseases and having a very good life, a good and you can say very fruitful life. So the knowledge you have shared with us, I think everybody would have enjoyed this one, and I would like the audience, if they have any query in this regard, if they want to pose their questions, any query in this regard, please they come forward and share their queries. I think Dr. Ravi will be there with us to solve your queries. Any query from any side? If anybody is having any query, otherwise we can do one thing, that if you have any problem, we can share the contact number of Dr. Ravi, and we can share each other the mail IDs from either side, and we can have their solution later on also. If you feel that you are having queries, if you feel some sort of questions raising later on in your mind. Any question rather now this time, you can call one. I think everybody is having, maybe possible that they must be calculating the issues, and hopefully if they would be having any query, they would be there with their questions later on. They can share their queries with me, and I will forward these queries to Dr. Ravi, and we will let them sort it out later on also. So I think with this we come to the concluding session now. Dr. Harish is there? Is Dr. Harish Singhal is there? I think he is busy somewhere else. Okay. Now I come with the acknowledgement, the vote of thanks for this program. I very much acknowledge my deepest gratitude to Dr. Ravi sir. Though we have been planning this lecture well before, but due to some engagements of Dr. Ravi, because he has to travel to Australia for almost 1 month for some academic engagement, and that's why he wouldn't. Earlier we have planned to arrange this lecture in May as well, but we couldn't manage because he has an opportunity to travel to Australia. This way we couldn't arrange that time. But his kind gesture is with us, and he told that whenever I will be back, I will be sharing my lecture with all of you, and he was very keen inside. And as soon as he returned back, he contacted us and told us to turn up with the appointment. And today that assignment was done, and I am very much thankful to him that he has kept his voice, and he has shared his extreme of his knowledge with all of us. Thank you very much, Dr. Ravi sir. And I would also like to thank our Honorable Vice Chancellor sir, that he always motivates us for such type of activities, because the academic institutions are supposed to nourish the fraternity, whether it is Ayurveda fraternity or modern fraternity. Each and everybody is supposed to be nourished by the knowledge, one step ahead. Whatever is being studied in current scenario, and what the data structures are being available in the modern scenario, this should be shared with all the fraternity, so that they get updated with the knowledge. So that's why our Honorable Vice Chancellor always motivates us to organize such type of activities. And this program, this third lecture this series was also planned on the directions of his Honorable sir, and this third lecture was also organized in the same way. And further onwards, I would like to acknowledge my deepest gratitude to the dignitaries, to the personalities present here across India, including the university. I would like to thank my deepest gratitude to Dr. Nilesh Borde from Goa, Dr. Prahlad Raghu from Kaskat, UP, Dr. Sachin Sharma from Kurukshetra, Dr. Sachin Sharma from Punjab, Dr. Jitendra Kumar from Udaipur, Professor Neelam Gupta, Principal Ayurveda College, Government Ayurveda College, Varanasi, Dr. Namrata Tiwari from Raipur, Dr. Yashpal from Jaipur, and moreover, Dr. Saroj Patil from Velak Ayurveda College, Pune. And in addition, maybe possible some of the persons I would not be able to recognize. I would not like to acknowledge all the dignitaries who were present in this lecture from across India. And I would also like to thank our principal, Dr. Mahen Sharma sir. He was our Professor Mahen Sharma sir. He was also present in this lecture. Dr. Dinesh Sharma, Dr. Hari Singhal, my co-organizing member, Dr. Ritu Kapoor, Dr. Govind Gupta, Dr. Nidhi Vaspi, and Dr. Prem Prakash Vyas sir, and other persons, all the PG students, all the PG scholars, all the PhD scholars from either side, either from the IIT or from the Ayurveda fraternity, and each and everybody, I would like to thank for their valuable time sparing in this lecture. Thanks once again to each and everybody, and hopefully we will be organizing a fresh lecture again, and the dates and time will be announced accordingly. With this, and especially I would like to thank the IT team, whole IT team who has been engaged in this program, all the employees of the IT team, because it wouldn't have been possible without their support, and especially Dr. Hari Singhal sir. Thank you very much, and And I hope with this we conclude this session. Thank you once again to everybody, and thanks Dr. Vankayala sir. Thanks for your.
Thank you so much, Rakesh ji. Thank you so much. Namaskar. Namaskar.
We'll meet again. Okay. Why did you stop from here?